Hello students, welcome to Read Med Prep Academy channel. Today in plant tissue culture part 2, we are going to discuss about the plant regeneration pathway, applications of plant tissue culture and conservation of plant genetic resources. Regeneration of plants. For the explants, plants can be regenerated by somatic embryogenesis or organogenesis. Here you can see the flow chart of the plant regeneration pathway. An explant is taken. The explant can undergo a direct embryogenesis resulting in the formation of embryoids or plantlets, and hardening is done of the plantlets, and these plantlets are transferred to the field for commercial production. Or the explants can be used as a meristem or a shoot apex, where this helps in the formation of virus-free plantlets. The stem or a part of the node can be used for multiple shoot induction for micropropagation, and this also helps in root induction, formation of plantlets, and hardening is done, and this is transferred to the field for commercial production. And the explants can be in the form of a leaf or a root, or the explants develop into a callus. This callus results in the formation of an indirect embryogenesis. This helps in the formation of shoot induction or root induction and results in the development of small plantlets, and these plantlets undergo hardening and they are transferred to the field for commercial production of plants. Here in this you can see another pathway where the explants by direct somatic embryogenesis can occur, and they also can be used for the production of callus resulting in the formation of somatic embryo, or by indirect organogenesis that helps in the formation of root organogenesis or shoot organogenesis, and this root or shoot organogenesis results in rooting and development of small plantlets. The somatic embryo can also be used by indirect somatic embryogenesis to result in the development of plantlets. By direct organogenesis of the explants, rooting can be done resulting in the formation of the plantlets, or the plantlets can be used for formation of explants, which can be used again in the cycle for development of new plantlets. Now, what is this somatic embryogenesis? Somatic embryogenesis is the formation of the embryos from the callus tissue directly, and these embryos are called embryoids, or from the in vitro cells directly form pre-embryonic cells which differentiate into embryoids. In this you can see how the somatic embryogenesis is done, where the male and the female gametes are made to fuse to form a zygote, and this zygote slowly enlarges to form an elongated zygote and a 2-cell pro-embryo and a 8-cell and then 16-cell embryo, and results in the formation of a globular embryo, and this forms the heart-shaped embryo. This is how the embryogenesis is done, which helps the development of seed production as well as for the formation of embryoids. What are the applications of somatic embryogenesis? This provides plantlets formation, which after hardening period can establish into plants. Somatic embryos can also be used for the production of synthetic seeds. Somatic embryogenesis is now reported in many plants such as Allium sativum, Hordeum vulgare, Oryza sativa, Z-maize, and this possible in any plant. Synthetic seeds are produced by encapsulation of the embryoids in agarose gel or calcium alginate. Here you can see the procedure for the synthetic seed production, where there is an establishment of the callus culture and induction of the somatic embryogenesis in the callus culture, maturation of the somatic embryos, encapsulation of the somatic embryos. After encapsulation, the artificial seeds are tested by 2 steps: test for embryoid to plant conversion and greenhouse and field planting. Organogenesis, the morphological changes that occur in the callus leading to the formation of the shoot and the roots, is called organogenesis. you can see in the flow diagram, the callus is differentiated into the shoots, and the development of the shoots is called cotylogenesis, and the development of the roots is called rhizogenesis, which constitute for the development of a good plantlet and later into a mature plant. Induction of organogenesis can be done in vitro by introducing the plant growth regulators in the MS medium. Auxin and cytokinins induce the shoot and the root formation. What are the applications of plant tissue culture? Development of the hybrids and the synthetic seeds. Improved hybrid production through somatic hybridization. Somatic embryoids can be encapsulated into synthetic seeds. These encapsulated seeds or synthetic seeds help in conservation of the plant biodiversity. It also helps in the production of the stress-resistant plants like herbicide-tolerant plants, insect-resistant plants, virus-tolerant plants, heat-tolerant plants. Micropropagation technique to obtain large number of plantlets of both crop and tree species is useful in the development of forestry within a short span of time and all through the year. Now, production of secondary metabolites from the cell culture utilized in pharmaceutical, cosmetic and food industries. What is somatoclonal variation? Somatic variations found in plants regenerated in vitro, that is variations found in leaf, stem, root, tuber or propagule. Gametoclonal variations found in plants regenerated in vitro gametic origin, that is variations found in gametes and gametophytes. Here the micropropagation of plants are done at the industrial level. In banana, it maintains high standards of homogeneity in plants like pineapple, banana, strawberry and potato. Here in this image you can see the banana plantlets being grown in vitro culture. Now, artificial seeds are synthetic seeds, otherwise called syn seeds. They are produced by using the embryoids or the somatic embryos obtained through in vitro culture. They may even be derived single cells from any part of the plant that later divide to form cell mass containing a dense cytoplasm, large nucleus, starch grains, proteins and oils, etc. To prepare the artificial seeds, different inert materials are used for coating the somatic embryoid like agarose and sodium alginate. Here you can see the artificial seeds where it has an artificial seed coat, an artificial endosperm and a somatic embryo. What are the advantages of artificial seeds? They have many advantages over the true seeds. Millions of artificial seeds can be produced at any time at low cost. They provide an easy method to produce genetically engineered plants with desirable traits. It is easy to test the genotype of the plants. They can be potentially stored for a long time under cryopreservation method. Artificial seeds produce identical plants. The period of dormancy of artificial seeds is greatly reduced. Hence, growth is faster with a shortened life cycle. Virus-free plants can be grown like the perennial crops, usually infected by variety of pathogens like fungi, bacteria, mycoplasma, viruses, which cause considerable economic losses. Chemical methods can also be used to control fungal and bacterial pathogens, but not viruses generally. So, a short meristem tip culture is the method to produce virus-free plants because the shoot meristem tip is always free from viruses. Here you can see the shoot tip or the apical meristem, which is sterile and free from viruses. This can be used for the propagation of the plants. Now, let us move on to the conservation of plant genetic resources. There are two types: germplasm conservation like the seed bank and cryopreservation. Germplasm conservation refers to the conservation of the living genetic resources like pollen, seeds or tissue of plant material maintained for the purpose of selective plant breeding, preservation in live conditions and used for many research works. Germplasm conservation resources is a part of collection of seeds and pollen that are stored in seed or pollen banks so as to maintain their viability and fertility for any later use such as hybridization, crop improvement. Germplasm conservation may also involve a gene bank. DNA bank of elite breeding lines of plant resources for the maintenance of biological diversity and also for food security. Here you can see multiple test tubes with different types of seeds being stored in the seed bank. Now, what is cryopreservation, which is done at minus 195 degrees centigrade? It is also known as cryoconservation. It is a process by which the protoplasm, cells, tissues, organelles, organs, extracellular matrix, enzymes or any other biological materials are subjected to preservation by cooling to very low temperatures of minus 195 to 196 degrees centigrade using liquid nitrogen. At this extreme low temperature, any enzymatic or chemical activity of the biological material will be totally stopped, and this leads to preservation of the material in the dormant status. Later, these materials can be activated by bringing to room temperature slowly for any experimental work. Here you can see the small test tube being stored by cryopreservation with the essential plant material. Protective agents like dimethyl sulfoxide, glycerol or sucrose are added before cryopreservation process. These protective agents are called cryoprotectants since they protect the cells or tissues from the stress of freezing temperature. So today in plant tissue culture part 2, we discussed about the plant regeneration pathway, applications of plant tissue culture, conservation of plant genetic resources. Thank you. Kindly subscribe, like, share and comment to channel Read Med Prep Academy. Log on to www.readmedprepacademy.com. Our Facebook ID is Read Med Prep Academy. Our email is readmedprepacademy@gmail.com. Our Instagram is Read Med Prep Academy. Kindly post your questions in the comment box. We will reply with appropriate answers. Thank you very much.